Wire at Berkeley at the Museum of Vertebrate Zoology. 3 years as a postdoc at the American Museum of Natural History with Frank Burbrink. And today he's an assistant professor at the University of Kentucky. So we met there while I was doing my postdoc. One of the things that I really enjoy about Skip is you have a real infectious enthusiasm for all animals, but herps in particular. And he's done some really cool work in Madagascar on gym snakes and on gecko diversification. So you hear about some of that today. Also, your enthusiasm extends into your hobbies. Skip is an amazing 3D modeler, 3D printer of herps, dinosaurs, I guess, exclusively. So
You're really outing me right now.
Instagram at PaleoSkip, or if you want his wildlife photography handle, it's SquamAteSkip. I'll leave it there. So we're gonna go to Bombs Away after this. Anyone is welcome to join us, especially if you have more questions for Skip. So I'll leave it there.
You can say Skip one more time. Thank you, Katie. You guys can hear me just fine? Yeah, so today is going to be also thank you for everybody that I got to meet today. This is a really lovely department. So this is going to be a pretty wide-ranging talk. I'm going to be talking about two and a half different systems that I've been working on since my PhD. And I am by training a molecular phylogeneticist and a comparative phylogeneticist, trying to dip my toes into genomics. So one of the things that's the uniting theme of my research is understanding the asymmetry and biodiversity across clades in deep time. So you'll notice that some clades are, even just on a precursory glance, that some clades are really diverse in terms of species and some are very depauperate, right? And there are lots of mechanisms driving this asymmetry and diversity. So here's just a simplified phylogeny. In this case, I think they're using genera or the number of families. This is from old paper from the 80s from Benton, where the width on the x-axis corresponds to the number of families in a clade. It's an arbitrary unit of measure that's really common in paleontology. And also on the y-axis is time, where you get towards the present as you move up. And you'll see that some clades are incredibly diverse relative to their nearest sister taxon, and that in some clades, diversity in the fossil record is heterogeneous. There's going to be a lot of me walking back and forth. So one of the things that I'm really interested in is how this comes about, particularly in groups that we really don't have a good record of the fossil record. I mean, I have a particularly good fossil record, which is everything I've ever worked on. So we're relying on basically molecular data to tell us something about the past. And the clade that I have conducted most of my graduate research on and postdoc work on, and that I'm working on in my lab now, are lepidosaurs. So lepidosaurs are all snakes, all lizards. They belong within this clade. And there are two major orders within Lepidosauria. The one in the red box is the sole surviving member of Rhynchocephalia, right? So that's the tuatara from New Zealand, and everything else on this page is a squamate, which is a sister taxon of Rhynchocephalia. And there are over 11,000 species of squamate. There are more species of squamate than there are birds. So this is the most diverse clade of amniotes or tetrapods in the world. And this includes all the snakes, things that look like snakes that are not snakes, amphisbaenians, pythons, geckos, gamete lizards like the frilled lizard, things that are related to skinks, chameleons, iguanas. So it's just an incredibly diverse clade. But what's really interesting is that when we look at the fossil record, even though it's lousy for both of these groups, that it seems that there was a flip around the KT boundary, where during the time of the dinosaurs, Rhynchocephalians were very, very diverse, and squamates were actually depauperate. And I followed the KT boundary where we had this turnover event, right? So even though I don't specifically study this particular turnover event, it's an example of something that I'm just really interested in. So the way this talk is going to be structured is I'm also a very fast talker, so I apologize in advance. I'm going to talk a little bit about phylogenomics and biogeography, and then I'm going to talk about something that my lab really focuses on. This is the phenomenon of adaptive radiation, which is something which may be familiar to some of you. So I am particularly interested in, or I started off being really interested in building phylogenies. I'm more interested in doing interesting quantitative things with phylogenies to answer specific questions. But one of the groups that I've been, one of all the squamates I'm particularly interested in, I am particularly attached to geckos. So geckos, if you're not familiar with geckos, geckos are just one group of lizards. They are effectively the sister taxon of all other lizards. And this is a divergence that goes back about 220 million years. And they're weird in a number of ways. If you guys have done field work in the tropics or stay in the tropics and you've had some little lizard running around on your walls at night, chances are it was a gecko. And they have a number of morphological adaptations. They're only primarily nocturnal group of squamates. Squamates are diurnal animals for the most part, with the exception of snakes and some fossorial things. And geckos correspond to about 25% of all lizard diversity. So there are about 2,200 plus species of gecko globally. They're circumtropically distributed and they're incredibly diverse in morphology. So for my dissertation, I was particularly interested in one clade of gecko called the diplodactyloid geckos of Australasia. And if you think about the gecko phylogeny, there are two major clades, one of which diplodactyloids represent, right? And they are endemic to Australasia, while the other gecko lineage is circumtropically distributed. Okay, so what's really cool is that this split goes all the way back to like the early Cretaceous or late Triassic. So this is a really ancient split within squamates, right? There are 7 gecko families total, 3 of which belong to diplodactyloids, or about 230 species. Like I've said before, that they're endemic to the Australasian region, and this is East Gondwana. So we're talking about New Guinea, Australia, some of the oceanic islands in the Pacific, not necessarily parts of the Sahel Shelf like parts of Indonesia and Malaysia. And what really drew me to this group is that it's a really charismatic group that has some really weird phenotypes, but the phylogenetic relationships were not well resolved. So I don't know if I can actually advance slides with this. I cannot. Oh, I can. Okay, cool. Right. This makes things way easier. So the 3 families are all morphologically very, very distinct. There are 2 families that have fully limbed members. They all have 5 digits on each toe, and they all have 4 limbs. The Carphodactylids lack toe pads that characterize a lot of geckos. About a third of gecko species in the world do not have any adhesive toe pads. And it's a really labile trait that's evolved and been lost multiple times in geckos. And Carphodactylids, a lot of them have really unique tail shapes, like this little nasty little dongle thing here on their first, right? So we still don't know what they do with this tail, other than the fact that it's packed full of sensory organs, right? This is Saltuarius. It's convergent with Europlatus in Madagascar, so leaf-tailed geckos, the classical leaf-tailed geckos. This genus Carphodactylus. These guys, when their tails, they automatize their tails, they drop their tails under trauma. And the tail itself actually makes noise to distract the predator. We don't understand the mechanism of how they do this. The other limb group is the more diverse group. There are about 35, 40 species of Carphodactylid, and there's like over 180 species of Diplodactylid. And Diplodactylids are way more widespread. They're found all over the South Pacific. And they are really diverse. Some of them are really popular in the pet trade, but some of these guys, like Strophaurus, they actually have a mechanism in their tail. A general theme is that they have weird tails. These guys, Strophaurus, there are 2 species of Strophaurus that will actually shoot a sticky white noxious substance out of their tails that seems to have evolved for deterring arthropod predators, right? Some of the largest and smallest geckos in the world belong to this clade. Geckos are typically smallish lizards, but some of them can be as big as my forearm. Some of them specialize in eating other lizards. Others are like rock crab specialists. Some of them specialize in eating like termites and stuff. Some of them are diurnal. And then the last group are the Pygopods, which are probably the craziest things out there in terms of geckos. These guys are near limbless, so they completely lack a pectoral girdle, and they have a partial vestigial pelvic girdle. Their common names are called flat-footed lizards because their little femurs stick out and they flap them around when they're trying to get around. And these guys are like almost all tail. They look like snakes, but they're not snakes. Most of them have external ears too. So it's an easy way to distinguish them. So you can see that this is a very diverse group, at least from my perspective as a herpetologist, compared to other lizards. And just as an example, is that geckos, not only is their postcranially diverse, their cranium is really diverse. That corresponds with trophic niches, right? So this is a Pygopodid lyalis. You can see the external ear opening here, eating what I think is a house gecko. And these geckos are defined by having a frontal parietal suture that allows cranial kinesis, dorsal ventral cranial kinesis, so the skull can flex, and it's extremely pronounced in this genus Lyalis. They also have hinged teeth, right? So these guys specialize in eating other lizards. They exclusively eat other lizards. And they have lots of adaptations for allowing them to grab slippery prey without them losing because they don't have limbs, and also wrapping their heads around it. So cranial kinesis is an ancestral condition for squamates, but different lineages, including snakes, have gone off and done wild things with having super flexible skulls. These guys get quite big. They get like 3 feet long. So this will be the last general diversity slide I have. So diplodactyloids are incredibly diverse, not only in body shape, but body size. So some of them, like Crenidactylus, are about the size of a quarter, whereas the largest living gecko is the giant gecko of New Caledonia. These guys, you can guess this is my silhouette here is, big smooth head and wide shoulders. So these guys are as long as my forearm, so these guys are huge. So there's about a 3 order of magnitude range of body sizes in this clade. And the largest geckos that we know about that have ever lived are also part of this radiation. So this is a species that was originally thought to be from New Zealand, although we don't quite know from its exact provenance. It's from the early 1800s. And here is a picture of that actual skin. It's known for a single skin that with bones in it. It's actually a mostly intact skeleton inside the skin with really crappy field notes associated with it. And this is at the Paris Museum, right? I'm not going to talk about, there's actually been some cool research on the phylogenetics of this particular gecko. And it turns out it's not related to what we thought it was related to. And then we have a limbless guy. So Lialis is the giant one down here. So these guys were found exclusively in Australasia and the South Pacific. So the bulk of diversity, about 2/3 of all diversity, is found on mainland Australia. There's a single species that's made it to New Guinea. And then there are 2 islands off the coast of Australia, one of which you've probably heard of before, New Zealand, right? And the other is New Caledonia. And what makes these islands special is that they're part of a sunken subcontinent called Tasmantis or Zealandia. So Zealandia separated from Eastern Australia during the tail end of the Gondwanan separation event during the late Cretaceous. So about 80 million years ago, Zealandia spread eastward, and both of these archipelagos have incredibly high endemism. And New Caledonia floral endemism is something like 80%. Like the only place in the world that has higher floral endemism is like Madagascar, right? And one of the early biogeographers made the argument that these Tasmantis lineages must be ancient relics from Australia, that this ancient Vicariance event carried with it the biota, a subset of the Australian biota, and they became endemic to isolated on New Caledonia, New Zealand, respectively. And this pattern this hypothesis was pretty wholesale across different lineages, including plants and birds and stuff like that. So there's lots of flightless birds on both archipelagos, and it's often assumed that the biotas of New Caledonia and New Zealand are related, right? More recent molecular evidence suggests that New Caledonia was actually underwater until about 37 million years ago. So Tasmantis, this giant landmass, drifted east and then was submerged, right? New Zealand had a lot of recent orogenesis, mountain building, and it's unclear if this is it was ever really completely submerged. Whereas in New Caledonia, it's pretty widely accepted that it is. And I should also note that geologically, New Caledonia and New Zealand are really distinct from islands. You can't see it here, but there's an island chain over here called Vanuatu, and further east there's Fiji, and there's New Guinea and the Solomon Islands over here. Geologically, they're very, very different. These are part of continental crust, so these are continental islands, whereas parts of New Guinea and Vanuatu, Fiji, the Solomon Islands, these are volcanic islands. They came up from the seafloor. Okay, now I keep going back to the computer. So what I was really interested in is I'm just going to fly through the phylogenetic stuff because it's really boring and no one cares. But what I was really interested in, and I didn't really have a big preamble about this to save time, is building a phylogenomic estimate of this particular clade of geckos because they are really interesting in terms of biogeography and macroevolutionary studies. People have these very strong opinions on where they came from and who's related to who based off of morphology, and with no real strong consensus. So I went about sequencing ultra-conserved elements, these parts of the vertebrate genome, or just animal genomes in general, that are more or less conserved. So if you look at the genome, the core of these loci, these regions, they might span into multiple genes, are very, very conserved. And as you get farther away from that conserved element, the more variation you get in those elements, right? In those loci. And it's that variation that tells you something hopefully interesting about the evolution of these lineages, if that makes sense. So just like for comparison, one of my earliest papers, I built a phylogeny for just a small group of these guys, a subset of these guys from New Caledonia, and I had 6 loci. For my PhD, I targeted 4,200 loci or 5,000 loci, captured 4,200. And one of the issues you have with phylogenomic data sets, I don't care what loci or locus set you're looking at, there are many, many different probe sets you can choose from, is if I take all this loci, am I actually increasing my power of phylogenetic resolution, or am I just adding a bunch of noise? Am I getting 5,000 different trees that don't tell us anything about the evolutionary history of this clade? And I think is cool is that if you actually look at individual gene trees made or species trees made from subsets of the 4,200 loci, we actually find that you only really need about 1,000 loci to recover the topology of the full tree. So there's actually a lot of power in recovering relationships in small subsets of this data set. We also find, I'm not showing it here, but we also find that variance between individual UCE loci is actually relatively low, right? Even, which is actually surprising because if you think this is not exon capture, in exon capture, you're targeting only exonic regions of the genome. And here you might be capturing parts of regulatory elements, introns, exons, all sorts of stuff, right? So it isn't really specific to a functional region of the genome. And what's really cool is when I started this project, I was told I was wasting my time because it's just going to be the mitochondrial tree. And it turns out it's actually not the case. So at the time, this is the most comprehensive phylogeny for this clade. We were able to recover like 95% of all relationships with high statistical support. The topology was novel, so it was really different from previously published mitochondrial phylogenies. And that if you actually look at Robinson fold distance or other tree-to-tree distances, this is calculating the actual number of shared nodes, portion of shared nodes across phylogenies. And we actually find there's actually really low overlap. So from previous studies that was claimed, lots of people have worked on this using Sanger data, that we find that the overlap as at maximum around 60%. Right. So the topology of this clade, the statistical support that we find per node for this study, I have two phylogenies from my specific study, vastly exceeds that from other studies, particularly super tree studies. I'm not going to talk about the details of it. So the key point here is that my tree is more robust statistically than smaller Sanger locus data sets. Also, and when we calculate the differences between my trees and the topology of other published studies, we find that there's a really wide variance. So they agree on anywhere between or disagree on anywhere between 36 and 40-something% of relationships, right? So not the key point here is that the topology of this from my UCE study is it's a new topology, and it's novel and it's statistically supported, right? Which is again kind of boring, but really important for doing this stuff I want to do downstream with this group, which I'm really interested in. Say, how do we get giants? How do we get miniaturized guys? What sort of patterns can we actually quantify in this clade? It's really dim. So I'm really interested in the phenomenon of adaptive radiations. Just in a nutshell, I'm not going to go into all the history of it, but adaptive radiations are lineages from which you have diversification from a single common ancestor, primarily along ecological axes. Okay, so this leads to phenotypic and niche phenotypic differentiation and expansion of niche space, or at least changes in niche space, right, within lineages. And you can think of like, well, that kind of makes sense. That describes basically everything, right? But it's not necessarily true because a lot of the lineages that we see today will also fall on this other type of radiation called non-adaptive radiation, where you can have extensive speciation, but taxa are more or less ecologically analogous, right? So there's less selection pressure for these things to diverge in terms of morphospace and ecology. And just in this cartoon here, in a non-adaptive radiation from a common ancestor, these guys look mostly the same. These different species look the same. Whereas in adaptive radiation, you have all these different shapes and sizes corresponding to ecology. And particularly, this relies heavily on the idea that shape dictates function in these clades. That ecology predicts shape. And classic examples of adaptive radiations would be like greater Antillean anolis lizards or cichlid fishes, where you have species that live in close proximity to physiogeographic proximity to each other, but have mostly non-overlapping niches. So there are lots of, adaptive radiation theory is something that's like pretty political. People have pretty pyroclastic arguments about what constitutes an adaptive radiation versus what's not an adaptive radiation. I think a lot of that's kind of pedantic, right? But some of the expectations of adaptive radiations are that you would expect heterogeneous diversification rates relative to niche availability. So as niche space fills for any given region, there'll be a drop off and species accumulation and also phenotypic diversification. Phenotypic diversification is predicted to be correlated to niche availability, and that there's also a correlation between the phenotypic diversification and speciation, right? Specifically speciation, which plays into net diversification, right? So just to show this, and when we think about the accumulation of lineages through time, where X is where the X-axis is time and Y-axis is the number of lineages, you can superimpose a phylogeny here. In a non-adaptive radiation, or in a radiation that is or a lineage that's specifically not diversifying along ecological axes, you expect, this is on a log scale, by the way, you expect mostly exponential increase in the number of lineages. In the case of an adaptive radiation, in theory, you expect initially rapid speciation, followed by a plateau as niche space was filled. This is analogous to, I'm drawing a blank now, ecologist, carrying capacity in ecology. I'm drawing a blank. I can't remember his name. But this is analogous to the carrying capacity in population biology, whereas you have the number of individuals in a system, they're consuming all these resources. There's a finite number of individuals that can occupy that particular space. In the case of macroevolution, we're thinking a finite number of species that can occupy the. This is garbage, right? So we actually, when we actually look at the math behind this, and I'm not a mathematician, but we actually look at the math behind this, this pattern is almost indistinguishable quantitatively from lots of other patterns that have nothing to do with adaptive evolution. So I'm not going to spend any time talking about carrying capacities. And this pattern is called diversity dependence. This is analogous to density dependence in population biology. So what I was really interested in, and this is something I just recently published on, is can we actually, how often do we actually see empirical adaptive radiation that fit any of these theoretical criteria, right? What is the relationship? Do we have predictive relationships between speciation and phenotypic diversification? Intuitively, that makes sense, right? You would expect that as lineages, as populations from a single common ancestor are become partitioned based off of niche space, that reduces exchange of alleles, increasing reproductive isolation, and you sort of have the snowballing effect. As lineages become specialized for something or generalized for something, they're less likely to interregress into adjacent populations. And that allows for sympatry, levels of sympatry that you often see in adaptive radiations. And the other thing is I'm really obsessed with islands. And there's this general, a lot of the adaptive radiations we think about in the literature, most of them are on islands. So we think of like Hawaiian silver sword plants, Galapagos finches, Galapagos tortoises, Madagascar, which Katie is familiar with. You know, Australia in itself is a continental island with all this wacky crap going on. So are islands radiation special? Do they occupy novel niche space? And are they distinct in terms of their diversification dynamics from their parental clade? That's what I'm really interested in. So when we actually look at this, when I start quantifying diversification dynamics using birth-to-death models, and I used a bunch of different birth-to-death models I'm not going to talk about in depth, one of the cool things that we find is that if you look at the whole clade of diploidactyloids, I'm not teasing out the island radiations or anything. And I had a priori hypothesized that the island radiations had a higher rate of speciation than the background rate for diploidactyloids. So the guys living on New Caledonia and New Zealand were diversifying faster than taxa on the mainland. Right, so that was my initial hypothesis. But when we look at the entire clade, we actually see a fair amount of heterogeneity in terms of net diversification. Right, this is a special metric called pooled diversification that I'm not going to go into explain, but this pattern is mostly similar to what you see with net diversification, which is speciation rate minus species extinction rate. And I should also note that that in my field of macroevolution, lots of these birth-death models try to estimate extinction, and it just can't be done from a molecular phylogeny. Molecular phylogenies for them in and of themselves are fairly depauperate in terms of what sort of information they can tell us about the deep past beyond relationships. So we have to do all sorts of shortcuts to figure out, like, was there a mass extinction event here? Do we see asymmetrical speciation in this clade versus another? So there are lots of models that try to do it. and the general consensus is none of them do it very well, right? So I'm still going to keep doing it. I don't know. But what we actually find is that when we look at net diversification, I'm sorry, pooled diversification, we find that early on in this clade's history that diversification seemed very, very stable with multiple jumps. This is from a paper I recently published. This one may or may not correlate with a global climate change event during the Eocene and Oligocene boundary called the cooling event, where there are mass extinction events across marine taxa and certain terrestrial taxa. This spike here is an artifact of the fact that we're actually seeing the clustering of all extant species at the tips, which is the nature of molecular phylogenies. The thing about molecular phylogenies is one of the reasons that they're misleading in terms of these models. They can be misleading as these models, is that molecular phylogenies are increasing in diversity as you get closer to the present because we're only sampling things that are extant, right? Does that make sense? And this is why the fossil record is really cool and why it sucks for me because there's a single fossil for this clade, right, that we know of, and we don't know where it is in the phylogeny. But with this, when we actually start teasing apart the different parameters of the pooled diversification rate, so I'm looking at pooled PSR, pooled speciation rate here, we actually see that it is really stable early on in the evolution of this clade with few spikes and relatively low speciation rate, suggesting that by proxy, extinction rate was actually driving this heterogeneity here. So this is something I'm pursuing in the future. We don't really have a good mechanistic explanation for this. And this particular metric for diversification is notoriously problematic and explained because it's not telling you something empirically about the biology of the organism that other net diversification models can. But what I was really interested in is are the island clades, the two island clades, New Caledonia and New Zealand, I touched on briefly, are they special? Do they have higher rates of speciation than the mainland radiation? And what we actually find is no matter how you try to splice the tree or whatever birth death model you're using, we actually find that the answer is no. So this is a cohort analysis or cohort plot where I'm looking at a sample from the analysis in BAM where I've basically pulled speciation rates out from the analysis from the posterior. So there's hundreds of thousands of samples from the posterior. This is a Bayesian analysis. And I'm contrasting them. So these are tip rates that we can have branch specific speciation rates. We can have node specific speciation rates. And then we can have tip specific speciation rates. And what we find is that when we do pairwise comparisons across tips, we find that it's homogeneous across the board. So i.e., the island radiations are diversifying, or in this case, speciating at the same rate as the background clade, which is not what you would expect under adaptive radiation theory. And when we look at some other models, this is from a model called CLADS, which looks at branch-specific speciation rates. When we start looking at all the branches at sub-10 specific clades, we find no significant difference from the posterior in particular clades. You can see I've split up the clades and color-coded them here. And all you need to get from this graph is that there's near total overlap in diversification rates for these clades. So the island guys, New Zealand and New Caledonia, basically show the same diversification trajectories as their mainland relatives. So this sort of flies in the face of what I was actually expecting to find. So I wanted to look at phenotype too. So I also do a lot of comparative phylogenetics with morphology. So I went around, in addition to doing field work in the South Pacific, I went around to different institutions, all these different institutions in the US and Australia and New Zealand, and measured about 1,100 specimens from this clade, corresponding to about 148 species, for 17 ecologically relevant traits. So these are traits that correspond to directly how organisms would be interacting with their environment. So like dimensions of the head, dimensions of the limbs, and the digits and stuff like that. So these are not diplodactyloid topads, but these are other gecko topads, and you can just see that there's a lot of diversity here. Hopefully that's obvious. So I was really interested in saying, do some clades have high rates of high or low rates of phenotypic diversification, and is phenotypic diversification correlated with speciation rate? And is phenotypic diversification wholesale? So if we see a shift in one particular clade for a particular trait, do we see shifts in other traits? Right, because a violation of that would suggest that there's some level of modularity, or at least low integration between traits. And what we find is that whereas speciation rate is homogeneous across this entire clade, this is actually a taxonomically complete tree. This is partially simulated and partially empirical. But when we start looking at individual traits, we find that the island guys are really exceptional in terms of their phenotypic diversification when we look at things like body size. So these little red arrows here correspond to shifts shifts that are above the background rate for various traits. So we have 2 shifts in body size, both of which are in the island. This is New Zealand in gray, New Caledonia in green here. So these guys respectively have rates of body size evolution that are an order of magnitude or 2 orders of magnitude greater than everything else in this tree, right? We also find that on the island of New Caledonia, there's one trait that really stands out, and that is the relative length of the forearm. And there are lots of mechanistic reasons why this might be true, because we know that limb length really dictates the width of, like, say, a branch a lizard will sit on. I'm not going to talk about that. That's really interesting. And then we see a decrease in the relative shape of the toe pads in this one Australian radiation, which is not surprising because these guys, it's genus Edora from mainland Australia. These guys really all sort of look the same. They vary in body size, but not much. And they all basically do the same. You either live on rocks or you live on trees in this particular clade. Whereas there's no exceptional diversification in phenotype when you look at the other traits that I was examining here. And what's really interesting here is that even though you see a lot of diversity in phenotype here, with the reason I was thinking that the island guys would have much higher rates of phenotypic diversification, if you look at some of these internodes here, they're really short. Okay, so this is also why I thought they would have higher rates of speciation, which we did not detect. But this is actually does agree with the fact that we're finding high rates of body size evolution and other traits in the island guys, but not their mainland taxa. Does agree somewhat with adaptive radiation theory. However, this trait diversification is not wholesale, right? What we're finding is we're finding them diversifying along one particular axis and not along others, which was a little surprising. So what I next wanted to do was quantify the amount of integration between traits, right? So if you think about this is like taking into account various aspects of development, but also things like allometry, right? Like if I evolve to get a big body size, does my head stay relatively large relative to my body, or stay the same size relative to my body, or does it change, right? And what we find is that there's covariance between all traits, right? So most traits you can expect a shift if there's a shift in an associated trait. But what's really cool is that for some traits, in this case, we have the correlation between the relative length of limbs, the forelimb, and the size of the head, is that they differ very strongly based on where you live. So island guys have much higher strength of integration versus the mainland taxa. And what this suggests is that there's potentially a higher constraint in these insular lineages relative to the mainland taxa, which actually kind of makes sense from a population genetics perspective, right? You would have a founder event with a truncated genetic diversity for whatever population or propagule gets to these islands. And I talked a lot about Tasmanthus, and I forgot to mention that when we actually look at molecular dating of the island guys, we actually find that they're really young. They're way younger than we would expect based off of vicariance events. So this is actually transoceanic overwater dispersal during the very earliest Neogene or late Paleogene, right? Which is impressive when you have a little lizard that's like this big dispersing across 2 miles of I'm sorry, 2 miles, 200 miles of sea, right? So talking about the Tasman Sea that separates Tasmanthus from Australia. So the take-home points from some of my gecko work is that speciation and phenotypic diversification seem to be decoupled, which is not what you would expect with adaptive radiations. Although this pattern is not unique to this clade, a lot of other clades where we've actually looked at this that I've not been involved with seem to find this pattern. That the diversification dynamics on islands are unexceptional, which says something about the life history of these taxa that's not obvious, right? Like something that you would think of as like maybe generation time with shift in lineages that have exceptional rates of diversification, say on islands, right? This is what we find like in anoles. Island anoles diversify way faster than mainland anoles, even though they're analogous in terms of the number of species. It should be said that of the 2,200 species of gecko, there's really strong selection for clutch size. So all geckos lay either 2 eggs or one egg. And if you lay one egg, that means you're really tiny, right? You have really small body size. So habitat partially shapes phenotype, and that most traits covary, suggesting that there's very strong integration between traits, a very strong selection on the entire organism itself, right? So, like if you contrast to, like, say, the next group I'm going to talk about, snakes. Snakes are a tube with a head. Geckos are obviously on the outside, seemingly more complex than that, right? So there's going to be limitations if you're going to have toe pads to climb or something like that. There's going to be constraints on, say, how big your toe pads could possibly be relative to your length or something like your limb length or something like that. How long your body can be relative to the size of your head. Does not say that there's not variation along these axes, but when we actually look at, say, the rate or the strength of covariation, it seems to be strongly biased towards the island taxa having higher constraint than the mainland taxa, which again makes sense from a population genetic standpoint. And what's really interesting is that we don't find wholesale shifts. Like I expected the New Caledonian guys, which are incredibly diverse ecologically, to have high rates of all sorts of phenotypic traits, right? But in reality, it's only a couple, suggesting that, again, supporting this idea that there's higher constraint for certain traits that may be an artifact of biomechanics, but also maybe an artifact of what they got to these islands with in terms of diversity. So one of my grad students also is crazy about geckos. This is my grad student, Tom Lau. He's now in his third year, I think. So I guess it's bad that I don't quite know. But he has been, handed him this project trying to build a super matrix of geckos, and he's hit the ground running on it. So to date, he's built the largest phylogeny of geckos using data mined resources. And what we find is that when we actually look across all geckos, so just that orient you to the gecko tree of life, because I know everybody really cares about that, is the Diplodactylids group I've been talking about until this point is only here. And you can see that the bulk of gecko diversity corresponds to this other major lineage called geckonoids. And these guys are found all over the world, right? So all the geckos that you see in the New World, they're all geckonoids. And they are really, really diverse and in terms of phenotype. But what we find is that when we look at speciation rates, it doesn't seem to be correlated with biogeography. So there seem to be and more has something to do with probably has something more to do with the life history of these individual clades. And Tom's looking into this now, so I'm presenting his data, and I'm doing the best I can right now. But the bulk of gecko diversity belongs to geckonoids. We see two shifts in terms of speciation, right? Once at the base, near the base of geckonoids, and then within this genus Cerdodactylus, which I've published on a couple times. And these guys, people who know anything about geckos, Cerdodactylus are weird because they're the most diverse group in terms of species of squamates. So there are like something like 350 species of Cerdodactylus that have been described to date. And if you look at made a list of all the lizards that are described every year, like 2/3 of them would be Cerdodactylus. So these guys are incredibly diverse, and they're found all over Southeast Asia and Australia. But when we look at shifts in body size evolution, what we actually find is we find significant shifts that are heavily biased towards insular clades, right? So when we look at shifts in body size, we find 22 shifts in body size, independent shifts in the rate of body size evolution. So either increases or decreases relative to the background rate of geckos in general, the global gecko radiation. And 3/4 of those correspond to island lineages. And just an additional quantification of this, Tom did this analysis in using stabilizing selection models. This is I can't remember exactly which OU model it is, but when we think about continuous trait evolution, Brownian motion is that there is, in theory, no bound on the evolution of a particular continuous trait, right? That as you get farther from the root of a tree, the higher variance increases. The alternative to that, Brownian motion is a special example of a model called OU, Ornstein-Uhlenbeck, where you can have variance as you get farther from the root, but there's selective optima for a particular trait. So that's why you don't have elephants that are the size of squirrels or something, right? There's something constraining the evolution of that trait. Selection is forcing this trait back towards the mean. And what we find is that when we actually quantify rates of body size evolution for different biogeographic categories, that island taxa have much higher rates of body size evolution versus the mainland, and taxa that occur on both islands in the mainland have even higher rates of body size evolution. And that they occupy distinct, these communities occupy distinct adaptive optima. If you're looking at adaptive landscape, this doesn't say anything about the size of those species, but if you're looking at adaptive landscape, insular species occupy one area, species that bridge the gap between mainland and islands occupy another area, and mainland taxa occupy a third, which is really cool. So we're writing this up right now. And back to the phylogeny again, when we start actually mapping the rate of body size evolution across the clade, across geckos, again, we find that most of the shifts that we detect in terms of body size, higher or lower body size rates of body size evolution, are clustered on islands. And not only that, but the magnitude of body size, the rate of body size evolution is really exaggerated on islands. So here's the same phylogeny I showed you 2 slides ago, but the branches have been, in other phylogeny, the branches are scaled to time. In this phylogeny, the branches are scaled towards the rate of stochastic body size evolution. So Brownian motion, this is variance sigma squared up here. And we can see that the New Caledonian guys I've talked about ad nauseum before have some of the highest rates of body size evolution compared to even some other geckonoids that also live on islands. Just to note that these guys have some of the biggest geckos in the world, or the biggest geckos in the world. This clade here, this is like a geckonoid radiation, Spherodactylus, have the smallest geckos in the world. So these guys are like, you could put one on a nickel, right? That's how tiny they are, right? In terms of body size. Some of the smallest, they compete with some other lizards for the smallest amniotes in terms of body size. Tom has also like totally taken over this project, and he's done a whole bunch of biogeographic analyses that I don't quite understand because I don't really do that sort of stuff. But what he's been really interested in, if you know anything about geckos, is that geckos are typically thought of as a Gondwanan radiation. They're mostly thought of as a southern continent origin group, where the bulk of the diversity are in fact in the continents of Africa and Australia. And that's secondarily invaded Laurasia, right? So these are the northern continents. Does everybody know what Gondwana and Laurasia are? Okay, so some people are shaking their heads. Gondwana would be when Pangaea split up. The first split was between Gondwana and Laurasia, where Laurasia is North America and Asia, where Gondwana is South America, Africa, Antarctica, and Australia. Okay. So what Tom has found is that these analyses are extremely sensitive to the placement of fossils. So totally ignoring the fossil record, you find a very strong argument for a Gondwanan origin. But when you start including fossils, which he did here, which was actually really genius of him from the fossil record, we don't have that many good crown gecko fossils. So these are stem geckos. When we start looking at the fossil record of geckos, it turns out some of the oldest fossils we have are not from the southern hemisphere, but from the northern hemisphere. Some of the oldest geckos that we have are from Central Asia. So what we find is when we actually look at the analysis that he reran with fossils, we find not only a different route of origin for geckos, so we find a route of origin somewhere in Central Asia, but that it totally changes the dynamic that I'm not going to go into the details of founder events within the clade of geckos, and also contractions and expansions, right? So if you totally ignore the fossil record, you find all sorts of crazy patterns, dozens. And so I think he got something like twice as many contraction and expansion events when you ignore the fossil record, and it was halved when you incorporate fossils into these analysis. And this is something that most empiricists, most neontologists are going to struggle with, because we just don't have, unless you work on mammals or certain groups of birds, you just aren't going to have a good fossil record. And he's the previous phylogeny I showed was about 700 species. He's more than doubled our sampling now. So he's built the largest gecko phylogeny that's ever been made, and incorporates about 3/4 of all gecko species. Yeah, so this is just, again, showing that we have really high confidence intervals for a split between not just Laurasia and Gondwana, but specifically Eurasia and Australasia. And this actually corresponds well with the basic gecko phylogeny, where we have the diplodactyloids, which are mostly endemic to which are entirely endemic to Australasia, and then the geckonoids, which are found west, right? So it seems like the common ancestor was actually possibly found in Laurasia instead of Gondwana, which is what has been argued for decades now. So now I'm going to transition to another aspect of adaptive radiations that's really common, is that the phenomenon of convergent evolution. In a nutshell, Convergent evolution is the evolution of similar phenotypes, presumably due to similar selective pressures and lineages that are not necessarily related that results in the independent evolution of similar phenotypes. An example of this would be flight in birds and bats and pterosaurs, right? But when we look at squamates are incredibly diverse. And for instance, we think of snakes being as like a very special case of legless lizards, which they are. But limb loss has evolved more than 24 times independently in lizards, right? So it's not they're not special in the fact that they lack limbs. It's how they've lost limbs and what they've done with their heads that make them morphologically special. And what I was really interested in doing this with quantifying, and this opens a question like in terms of then snakes, they seem to be a good system to really pick one trait that is obviously highly labile and is driven by ecology, and that is the head. So a snake is a tube with a head and some eyes, right? And that's obviously an oversimplification, but snakes are also really cool because snakes account for about a third of all lizard diversity. There are over 4,000 species of snake, and that most of that diversity is actually found in one clade called Colubrids. This includes all the vipers, all the rat snakes, all the cobras, whereas pythons and blind snakes are all part of another, are outside of this clade. And what's really cool is that snakes are really diverse. Lots of islands have snakes, but most island assemblages of snakes are actually conglomerate assemblages, where you have independent colonization events from mainlands from different lineages. In the case of just one example that I know of, and that is the Malagasy gem snakes from Madagascar, where both Katie and I have done some work, we actually find evidence for a really extensive adaptive radiation where there's been extensive in situ diversification on the island, not an assemblage that's come together independently from different dispersal events. So this clade is also really cool because it's really young. So there's over 100 species, only about 80 of which are described. This clade's about 22 million years ago. Seems to have dispersed from Eastern Africa at some point during the Miocene, or I guess late Oligocene. And what's really cool is that there's a ton of morphological diversity in this clade that seemingly overlaps with other snake assemblages in the US. The US is a great example, and North America is a great example of a really diverse snake fauna that is a conglomerate of many, many different radiations that have come from Asia and South America, right? So what I was really interested in is testing can we quantify convergence in this clade? But before that, does ecology predict shape? All right, so I'm using slightly different methods here. Can we actually quantify the amount of specifically modularity in the snake skull, and also the amount of integration between modules? You would think of the snake skulls being highly modular, which is what I hypothesized, and that integration between different modules was very, very low, right? And that what I was really interested in is if for those people who don't work on snakes, if you go, particularly in the tropics, if you go to another part of the world and you're working in the tropics, but also in non-tropical areas, you'll often see snakes that look like a dead ringer for a snake that's in a totally different part of the world, and also a different part of the snake phylogeny. And I'm going to show some good examples of that in a few slides. So I was really wanting to see, like, can we quantify that? Because convergent evolution is by its nature hierarchical, right? And I'm only looking at, say, functional convergence in this example. Right, this is just a phylogeny of the Malagasy gem snakes. The actual name is Pseudoxyrophids, and you can see that it's from the skull. They are incredibly diverse in terms of morphology, and these are just live images of these guys. So we have taxa that are like leaf litter specialists. We have taxa that are seemingly convergent with our heterodon here in the U.S. If you guys are from the West Coast, you don't know what heterodon is probably, but in central U.S. and the East Coast, we have very large, heavy-bodied snakes that specialize in eating toads. They have their own version of this in Madagascar, right? We also have the only good example of sexual dimorphism in a snake and a secondary sexual characteristic in this arboreal snake called a langaha. This is the female, little Pinocchio nose. I'm sorry, this is the male with the Pinocchio nose, and the female has this leaf nose. Camouflage probably plays a role in here, but we don't really know what they're doing with them. We do know that the males can wiggle their nose, which is pretty cute. So it's yeah. So this is incredibly diverse. No two genera look really the same in this clade, but we know they share a common ancestor. When we look at the osteology, this morphology, this diversity in the skull and the head shape is reflected in the osteology. So there are two basic tooth types. Epistoglyphus, where these guys have rear fangs, and then there are aglyphus snakes, which have no fangs in the back of the jaw, right? So what I was really interested in is using geometric morphometrics. I went from using linear external measurements to CT scanning to get good 3D models of the skull. I feel like I'm going to go over. Should I stop? Okay. I'm going to fly through. I'm just going to skip over the methods, right? So what I was really interested, I used multivariate analyses and linear models to test the relationship between the shape of the whole skull, specific elements of the skull, and aspects of their ecology. And what we find is that when you look at ecology, there's a very strong relationship between, say, the shape of the brain case, where the brain is, obviously, the neurocranium, and uh, what sort of substrate they're living in, right? Which suggests there are biomechanical constraints on how, like, say, if you're digging through soil versus navigating leaves and trees. We do not find a strong relationship when we think about different with tooth shape or tooth type, fang type, and skull shape, which is actually a little surprising, right? You would think that there'd be a very strong relationship between the type of tooth you have and the shape of your head that contains that tooth. And when we actually go back and look at some of the similar analyses I did for geckos, when we quantify modularity in the skull, but specifically looking at different elements of the skull, snake skulls are simplified in terms of the number of bones compared to other lizards, but they're very complex in terms of the relationship of these bones. Snakes, obviously, they can't unhinge their jaw, but a lot of the bones in the skull are capable of moving independently or as part of a system. Right. So what we find is that there's high signals of modularity. These are pairwise comparisons from a covariance analysis of elements of various elements. The ones with dots was where we find significant modularity, signals of modularity between these elements. So we see that the maxilla is not particularly is likely not part of the same module as, say, the ectopterygoid or the mandible in this case, which is not surprising, right? It's something that we find in other vertebrates. But when we start looking at other parts of the snake skull, I don't have time, but if I went back and looked at the snake skull, you'll see that there are all these floaty bits that are dangling at the bottom of the snake skull, right? It's obviously different than a mammal skull. So in the case of a mammal, it's going to be high integration between, say, the premaxilla, which bears our incisors, and our maxilla, which bears all our other teeth. We do not find that sort of integration in the snake skull. Tooth-bearing elements don't seem to be integrated with one another. The brain case is not integrated with the integrated with the brain. The tooth-carrying elements in the lower jaw seems to be entangled from everything else, right? This is not a pattern you find in most other, at least amniotes, right? So here's a Bayesian analysis where I've looked at the strength of integration between modules. So we're measuring two different things. This is measuring modularity. This is the strength of covariation between modules, right? And what we find is that it's also simultaneously measuring the rate. When we break down these groups based off for individual pairwise comparisons, I'm highlighting, say, the maxilla and the brain case, we find that there's no variation in the rate of evolution for individual traits when we look at, say, gild. But then when we look at the brain case for Mellicassez, it's not really obvious from this plot here. This is the strength of correlation on the off diagonal here. When we actually look at significant differences start quantifying significant differences between the strength of integration between modules, it seems to be highly dictated by gilds, which is actually not something I was expecting. I was expecting there to be a much stronger phylogenetic signal. And it seems that based off of where you're living and what you're eating really dictates these red arrows suggest significant or imply significant differences between, say, in this case, maxilla and brain case for different gilds. Does that make sense? So this is a really cool novel finding I was not expecting to find. I really don't have a good way of explaining it. It's something I'm still working on. I'm actually going to skip over this. I'm going so slow. One of the other things that was really interesting is that adaptive radiations are typically thought of as being distinct in terms of their trajectory, their macroevolutionary dynamics from their mainline clades, but also distinct in terms of the morphospace that they occupy, right? So I wanted to quantify that. And in a nutshell, when we look at the Malagasy gem snakes, right, we find that they are not exceptional. And in fact, they seem to overlap. These are highlighted here in green. When we actually look at shifts using stabilizing selection models, shifts in morphospace to new adaptive optima, we find that these guys are not special. We find no shifts in the Pseudoxyrophene snakes, but instead the shifts seem to be driven by really specialized snakes, say aquatic snakes or things like vipers, right? So when we look at things like vipers, which are highlighted here in blue, and cobra relatives, they have really specialized jaws for bearing fangs that you don't see in these guys. And what this also suggests is that even though these guys are a good example of an adaptive variation in terms of ecological differentiation amongst lineages, they seem to be replicating what we see in other snake assemblages elsewhere in the world, right? Just broad, if I were to plot a morphospace plot, there'd be broad overlap with other parts of the world. This is the last couple slides, I promise. When we actually start quantifying the strength of convergent evolution, this is actually a really fraught topic too, or a fraught field of study of macroevolution. It's really hard to do without like a priori bins of guilds. But we start looking at, say, vine snakes across the world. So Langahagen, that Pinocchio nose snake, Ichthyophis from Madagascar, they broadly overlap in morphospace with neotropical and Asian vine snakes. You can see these are 3 different vine snakes from 2 different families that look very, very similar. So this is a Hetula from Southeast Asia. This is Euromacer from the Dominican Republic. And this is Oxybelis from Costa Rica. And these guys all have really elongated, narrow skulls and huge eyes when you look at the CT scans. When we look at, say, hognose snakes, is the genus Liheterodon from Madagascar. This is a Heterodon from the US, and Xenodon from South America. They all have these upturned rostral scale, which presumably is reflected in the morphology of the premaxilla here. What we find is that they actually don't have very strong signals of convergent evolution. It seems that they've all gone different routes for getting the same shape of the overall shape of the head, which is, I'm not going to talk about it too much, but which seems indicative of this phenomenon called many-to-one mapping, where you can go different routes mostly to get to the same answer, which is really cool. Hasn't really been documented in snakes, although it's not surprising given that snake skulls are really modular. So when we actually look at this in terms of the specific elements, we actually find that the story is way more complicated. And I look at this graph now and I realize this is not helpful at all. But the elements that have asterisks for a particular clade means that we find significant signatures of convergent evolution for that particular element, for that particular like clade guild, right? So for instance, we find strong signatures of convergent evolution specifically for the palatine in aquatic snakes, right? Whereas we don't find strong signatures of convergent evolution in the pterygoid. And that doesn't make any sense because both of them are tooth-bearing elements at the roof, on the roof of the mouth, right? So I'm still trying to wrap my head around this mixed signal here. And we seem to find high significance and strength of convergent evolution in the neurocranium as opposed to, say, the trophic elements, which suggests that there's very strong selection on the sensory elements of the snake skull as opposed to the dietary, the trophic bones of the skull, which may be more driven by phylogenetic signal. That makes sense. So I'm going to stop there. And I just want to say that my lab is currently looking for bioinformatics postdocs because my lab is now transitioning to comparative genomics, and I have some really cool preliminary data with gecko whole genomes that I'm trying to turn into a whole grant. So thank you. Sorry I went over, but.